So thank you very much for joining us for Damian Bailey's GL Brown Prize Lecture, which we'll be having in just a minute. Before we start, I just wanted to do a tiny bit of housekeeping. So just to let you know that the webinar is being recorded. Please can you keep your microphones and videos off for the duration of the meeting. And what we're going to do is the session's going to run to be a 45 to 50-minute talk, and then there'll be about 10 minutes of Q&A at the end. So if you come up with a question during the talk, if you post it into the Q&A into the chat, then we'll take questions from that at the end, and then we'll know that they're there. And yes, I think that's all the housekeeping that we need to do. We'll address them in the Q&A portion of the lecture, but I'll be checking in on the chat and registering what people have asked during the presentation. So you can ask the questions at any point. So I think that's all the housekeeping. I'll just see if we've still got, I think we've settled down on people are in. So that's really great. So it's my absolute pleasure to introduce Damian Bailey today to give the Physiological Society's GL Brown Prize Lecture. So this is given annually to an excellent physiologist in order to stimulate an interest in physiology. And I'm really happy that it's Damian giving it today. There's so much I could say about how well he deserves this GL Brown Prize Lecture. So he's got an amazing academic track record. He's got a plethora of credits for his fantastic science communication. So he's going to give us a great lecture. And he's contributed so much to the Physiological Society, including as the current editor-in-chief of Experimental Physiology. And so I can go into much more detail, but he's asked us not to talk about all that at length. So I'll stop. And I'll just say, here he is, the excellent Damian Bailey, Professor of Physiology and Biochemistry at the University of South Wales. And I'm delighted to pass over to him to give the 2025 GL Brown Prize Lecture. Thank you so much, Damian.
Thanks very much, Catherine, and really appreciate the introduction. Very kind, and thanks to everyone for joining. I know it's busy to take an hour out of your days, and really appreciate it. So this is the man himself, Sir George Lindor Brown, 1903 to 1971. Really his MO was to try and speak to a younger audience to stimulate a broader interest, if you like, in physiology. And that's going to be the focus for today. He made fundamental discoveries about how nerves communicate with muscles, especially the role of acetylcholine in signal transmission at the neuromuscular junction. And I think what I like most about G.L. Brown is there are four real adjectives that really explain his personality: modest, inspirational, compassionate, and brilliant. And for those early career researchers, in fact, across the continuum of expertise, I think those are wonderful things that we should all aspire to. So, surviving space, pushing the brain's limits. And my primary goal today, as Catherine mentioned, is to stimulate a broader interest in physiology. So I'm not going to get stuck into the mechanistic weeds, if you like, of the science. I just want to give you a high-level overview of some of the work that we've been doing. And as I say, the aim is just to shine a light on physiology. It's a wonderful discipline. And again, I really appreciate the Physiological Society for giving me the opportunity to do so. First and foremost, I'd like to thank my team, the Neurovascular Research Laboratory. It's full of people that are way younger than me, and actually there are some people that are probably as old as I am, and they're a wonderful team, and a special thanks to all of them, because without them I certainly wouldn't be in the position I am today, speaking to you, and also to many, many collaborators, too many to mention, 182 in fact, that I've collaborated with over the years, and we'll speak a little bit about that later on. So I want to talk to you a little bit about the brain and oxygen. This is important. Extravehicular decompression in deep space missions. Hypoxia is incredibly relevant. And also gravity. We've evolved over 4 billion years to depend on 1G. And so when you pull gravity out of the equation, it causes really interesting things, as you can see Jessica Muir here on the space station. And it's that delicate balance, if you like, and integrated physiology can give us some insight into where that balance sits. So, setting the scene: oxygen, gravity, and evolution of us, the obligate neurobes. If we go back in time to 4 billion years ago, the atmospheric paleoatmospheric oxygen concentration, as you can see here, even throughout the boring billion, aptly named. So about 1 billion years ago, we then see this shoot upwards in the atmospheric oxygen concentration, the Cambrian explosion. Oxygen, through photosynthesizing cyanobacteria. These are two oxygen atoms coming together, and they share a covalent bond. Fascinating structure, an all-important structure. And over the Cambrian explosion, if we expand on the sudden rise in the atmospheric O2, you can see that with each and every major uptick in the atmospheric oxygen concentration, it coincided with the explosion of life and biota. The evolution of the heart, the four-chambered system, so that we can split and control where oxygen goes and regulate it because it's so important. And also the brain, which has become, as you'll see, very, very oxygen hungry. And that has implications in terms of our vulnerability when we reduce oxygen and when we reduce gravity to the brain. Gravity, that fundamental vector that we've all evolved under 1G, it's invisible, we don't see it, but it has huge implications when we pull gravity away. And you don't get to do photographs like this at sea level, of course, under 1G. And that's the fascinating thing with microgravity, is that you can do these types of things. And the circulation has evolved with that pulsatility, the all-important pulsatility, because of gravity. We pull gravity away, and it has an impact on our ability to get substrate, oxygen and glucose, to the brain. And that's really important. And the brain is turning over a huge amount of ATP, oxygen and glucose, to push these ions uphill. So the turnover of ATP is an extraordinary at rest, 282 billion molecules per minute, and oxygen and glucose, of course, to be able to support synaptic transmission. If you look across the continuum of diseases of the circulation, for example, stroke, sudden interruption of blood flow to the brain, Alzheimer's and depression, you can see here with these MR and with these PET images, you can see that they're unified by a gradual reduction in oxygen and substrate delivery across the brain. And so this is really important. And the take home here from this 7T DTI image of the brain is that our brains need oxygen and gravity. It's essential. And we can push the bioenergetic limits. This is a study entitled On the Acute Arrest of the Cerebral Circulation in Man in 1943. By far and away the most unethical study that I've seen in the literature, certainly one of the most unethical studies. And here, these are schizophrenic patients, no consent, of course, in this study. And a crowd cuff was placed across the neck and bilateral carotid arterial compression in order to reduce blood flow to the brain. And the clinicians were asking the question, what happens to the brain when we reduce blood flow to it? So an extraordinarily unethical experiment, of course. And this occurred for over 100 seconds. And what's fascinating about the brain is that its oxygen content, 30 nanomolar per milliliter. And if you look at the cerebral metabolic rate of oxygen, the oxygen consumption at 30 nanomolar per milliliter per second, you'll understand that its oxygen reserves, in terms of its aerobic reserves, are a meager 1 second. And if arterial PO2 drops from what your arterial PO2 is at the moment, 100 millimeters of mercury, to less than 16 millimeters of mercury, the critical boundary arterial PO2, you'll slip into unconsciousness. That's how important oxygen is. In terms of glucose, the substrate that oxygen burns, if we empty the glucose tanks, which takes about 3 to 10 minutes, when the arterial glucose concentration drops to 2.3 millimolar, we slip into a coma. We're not really clear in terms of the boundary thresholds of ATP. And of course, these are extraordinarily difficult experiments to do. We don't do these experiments today, of course, but the bioenergetic limits of the brain are very limited. Limited glycogen stores and next to no oxygen binding proteins, less than 1 nanomolar per milliliter of neuroglobin, for example. And there are limits, human limits. These are a snippet of three separate studies that highlight how we can actually push the brain to its limits in environmental extremes. The late John Ernsting had volunteers breathe pure nitrogen and demonstrated that within 16 seconds they fell unconscious. The arterial PO2, again, that boundary, 16 millimeters of mercury. The extreme Everest group at high altitude at 8,400 meters measured an arterial PO2 of 19 millimeters of mercury, and the climbers were all perfectly conscious as a result of acclimatization, but extraordinarily low PO2s. And work initiated by the late Chris Willey from Phil Ainsley's lab at UBC with single breath hold divers, apnea, with arterial PO2s as low as 23 millimeters of mercury, and if indeed less than 20 millimeters of mercury. And again, they remain conscious. So we can push the brain in ethically approved experiments in a ethically permissible manner, unlike, of course, the Red Wing study that I mentioned prior. And we can actually start to try to understand what are the mechanisms that allow the brain to cope with such incredible environmental extremes. I've kind of given this a gold, a silver, and a bronze award, if you like, in terms of how hard the brain is being pushed in these experiments. And of course, if we turn to nature, nature has coughed up some wonderful examples. It's had roughly 550 million years of vertebrate physiology to be able to evolve and to accommodate to some of these stresses. From the swimmers to the flyers to the burrowers, nature has produced some incredibly hypoxia, anoxia tolerant species. The platinum, the best one for me, is the common crucian carp. 100,000 minutes of pure anoxia, not a single whiff of oxygen to the brain, and not a single protein to suggest there's any evidence of damage to the neurovascular unit, to the brain. These are the superheroes, if you like. And I think we can learn an awful lot from comparative physiology, which is a fascinating science, to shed some really unique insight into the clinical physiology. And what unifies all of these species in terms of their ability to cope with these environmental extremes, this lack of oxygen, is a reduction in oxygen consumption in the brain. It's almost a cryogenic state. And as we'll come to learn, free radicals, reactive oxygen and reactive nitrogen species, are involved in driving some of these responses that switch oxygen consumption down, which would make intuitive sense, in order to protect the brain. It's a unifying neuroprotective adaptation. People are born and bred at high altitude, and we've worked in this area with colleagues. This is a paper from one of my current colleagues, a past PhD student, Ben Stacey, published in the Journal of Physiology, where we were asking the question, what is it that's different about people that are born and bred at high altitude? Part of a wider expedition with my colleague Phil Ainsley at UBC. And there are differences. There are molecular differences, there are hemodynamic differences, and there are clinical differences. So living at high altitude where you're exposed for generations to low oxygen levels drives adaptations. And there's also a genetic predisposition, a genetic contribution to this as well, which colleagues are working very hard to try to disassociate, one from environmental, one from genetic. Perhaps this is why the Faith Kipyegons, the Eliud Kipchoges, born and bred at moderate altitudes here, both from Kenya, are such incredibly accomplished distance runners, where oxygen delivery to the brain, of course, is a key limiting factor in terms of performance. That'll be a topic this time next year, I understand, by Professor Andy Jones. So watch out for that. So we've evolved to depend on this molecule, oxygen, these two oxygen atoms coming together, bounded by this covalent bond. But if we take a deep dive look into its molecular orbital structure of oxygen, each and every one of these arrows is an electron. And as you can see here, these electrons are paired up with opposite spin. But there are two electrons in the outer antibonding orbitals of oxygen that are unpaired. So oxygen is a free radical. It's a diradical, a double radical, if you like. And what oxygen likes to do is it wants to accept electrons in order to fully complement these orbitals. But the Pauli exclusion principle means that oxygen accepts each electron, not instantaneously, because they'd have the same spin as they move into those orbitals. So it reacts, it accepts these electrons one at a time. And that's very fortuitous for us, because if it wasn't for the Pauli exclusion principle, if these two electrons with the same spin number were accepted into those orbitals, we would combust spontaneously in the 20.93% oxygen that we're exposed to. So there's a real interesting evolutionary quantum quirk there. And as the oxygen levels during the Cambrian explosion increased, the molecular machinery became ever more sophisticated to be able to control for oxygen, because at high levels, because it's a radical, it is damaging, it is mutagenic, and it's toxic, which is, again, in and of itself fascinating. So as you can see, in hypoxia, where um, where there's less oxygen available, we see this unifying increase in reactive oxygen species, which knocks out prolyl hydroxylase, allows the HIF1 alpha complex to accumulate, and oxygen-sensitive genes, target genes, to increase, basically to increase oxygen delivery to the organ. So it's a really fascinating story that we've evolved to be entirely dependent on this toxic mutagenic gas in physiologically controlled amounts, but in pathological excess, it can kill us. And what's really fascinating for me, with a father who's a particle physicist, is that this is quantum physiology, in as much as this is a radical. It can act both as a particle and a wave. So these radicals can interact in terms of signaling by superposition. They can be in two places at the same time, entanglement. When we measure something, it's going to have an implication for something else, the measurement phenomenon. And tunneling, it can break through energy barriers that would otherwise seem impossible to do so. So the fact that these radicals are acting as both particles and waves potentially can lead to some really unique advances in this field, and something I've referred to as quantum physiology. So how does the brain sense oxygen and gravity? Well, to answer this question, I'm going to turn to a very special physiologist in our house. This is my son, Luca. He was named Luca not because he's the last universal common ancestor, but my wife likes the Italian name. So this is something that Luca's preparing at the moment. He understands, even though he's 13 years of age, it's publish or perish. This is an antioxidant. This is a free radical. He's drawn this for us. You can see it's lacking that electron in the outer antibonding orbital. And this sacrificial antioxidant, for example, it could be vitamin C, is donating an electron. It's stripped off its orbital by the free radical in order to stabilize itself. And these radicals traditionally have had a very bad press, because if you have this continual stripping of these electrons, you get what's called lipid peroxidation. And this can cause destabilization of the cell membranes, and it can cause damage to the neurovascular unit. So radicals have had a pretty bad press over the years. But what we're coming to understand is that in physiologically, albeit undefined concentrations, reactivities, these radicals can act as hormetic molecular switches. They're perfectly poised to act as molecular switches, hormetic switches that can drive neuroprotection, neuroadaptation. For example, a hydroxyl radical has got a half-life of a billionth of a second. Now, as a signaling molecule, that's very fast, and it's perfectly poised, but can also cause damage when in excess. So we use a technique to measure these radicals. This is electron paramagnetic resonance spectroscopy. So we don't rely on indirect distal biomarkers of free radical mediated activity or damage. We actually measure the paramagnetic species themselves. And the way we do this, we use different diamagnetic spin traps that react with these paramagnetic species to produce this resonance stabilized adduct. Here you can see different adducts, which are specific to different species of radical. Those of you that are tuning into this lecture at the moment, you could all be a different species of radical with different reactivities, different one electron reduction potentials. Some of you could be very reactive, very aggressive, where you strip that electron very, very quickly. Others, for example, could be like superoxide, a very domesticated radical involved in cell signaling. So we measure these radicals directly. We're also interested in measuring all of the vasoactive nitric oxide-based metabolites. Again, we do this with old-fashioned ozone-based reductive chemiluminescence. Here you can see one of my lab colleagues, Ben Stacey, injecting a plasma sample into the reagent, and we're measuring this at picomolar concentrations. So that's a billionth of a mole, so very, very small concentrations. And we measure all of the vasoactive moieties, which is really important because there's a raging controversy at the moment in terms of what happens to the brain if we generate radicals or if there is hypoxic vasodilation. Is it nitrite reduction that's driving this, or is it S-nitrosohemoglobin formation that's driving this? So we're able to measure all of these moieties in order to understand these various mechanisms. We lean on a variety of really interesting experimental models. And again, this is where combining adventure with science, I find, is so inspirational for the early career researchers. You've got to try and think of the models that, you can get ethical approval for, of course, but are quite challenging in some ways. And we take an integrative focus, just as Roy and Sherrington did. This is back in 1893 in the old pathological laboratory at Cambridge. And we make a whole range of measurements from the molecular through to the hemodynamic, through to the clinical, through to the structural functional, in order to try to understand how the brain responds to some of these environmental extremes. We lean on clinical models as well to provide important insight because of the translational relevance of some of the physiology that we're doing. Here, this is a carotid endarterectomy. You can see the carotid arterial bulb exposed here. We're sampling blood going into the brain, the carotid artery, and we're sampling blood coming out of the brain via the internal jugular vein. This is a laboratory experiment that we would typically perform. This is a jugular venous catheter placed in a participant. Here, we've got a femoral arterial catheter measuring what goes into skeletal muscle, a femoral venous catheter measuring what comes out of skeletal muscle. We can then use thermodilution to calculate flow across skeletal muscle. We've got an internal jugular venous catheter measuring blood leaving the brain, and a brachial arterial catheter measuring blood coming into the brain. So we know exactly what muscle's doing. We know exactly what the brain is doing. We can measure flow in the extra and intracranial circulation. We can collect our blood. We can spin trap and isolate the different radicals we're interested in, and perform a variety of measurements. We can exercise our volunteers. We can expose them to less oxygen. We can shift them gravitationally. We can tilt them. We can spin them. And then we can use Fick in order to measure whether or not radicals are being released from the brain or consumed from the brain, and how this maps on, how this aligns to changes in substrate delivery. This is an experiment in collaboration with Phil Ainsley at UBC, dynamic end-tidal forcing. We're manipulating and maintaining carbon dioxide levels whilst changing oxygen levels. Tony Dawkins is doing some cardiac function measures. Ryan Hoyland on the right is measuring blood flow to the brain, and there's synchronous arteriojugular venous sampling across the brain. And Dennis Wakeham in the background there is doing some additional hematological measurements. So these are the types of experiments that we're interested in to get us close to the organ of interest, the brain, and to use the best techniques to make the measurements that we're interested in. You can combine adventure with the science. We can go to high altitude, and we've had wonderful opportunities where we've been on multiple expeditions with colleagues and collaborators, and the Himalayas, to Chile, to Peru. And we've recently returned via the European Space Agency from Concordia in the Antarctic, in Antarctica. This is at roughly 3,800 meters. We were able to image the brains of the crew there using a low field hyperfine swoop. So we flew this out to Antarctica. It stayed there for a year, and every week we were able to image the crew sequentially and make a variety of measurements. This is with my colleague Peter Uhlenberg from the University of Munich, and that's going to be a fabulous experiment to analyze. Also in collaboration with Dr. Phil Ainsley and the late Chris uh, 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 here you can see some superhuman freedivers, some apneists. This is Bunamir Sobat, who's able to hold his breath and free dive here. This is him actually breaking a Guinness World Record. Um, this here, 11 minutes, 54 seconds on a single breath. I mean, it's absolutely fascinating when you do the oxygen depletion calculations and realize how hard they're pushing themselves to the limit. If you were to put Buddha on 100% oxygen, he could extend this breath hold to just over 24 and a half minutes in order to take that Guinness World Record. And of course, with our collaborators, we're able to instrument across the brain and to measure the free radical exchange kinetics across the brain during these types of maneuvers. An incredible challenge for the brain in terms of the hypoxia and also the relative hypercapnia associated with these single breath holds. Some more recent work that I'm engaged with at the moment with a visiting fellowship at Yale University in the US is the first isolated pulsatile brain system. And here, this was the original BrainX setup pioneered in the pig brain, the mammalian brain, and we've since shifted to the human brain. Here, the pig is sacrificed for food. The brain is the head is decapitated and it's typically thrown away. The brain here is surgically isolated. It takes about 6 hours to do so following decapitation, and it's placed on a pulsatile rig where we then reperfuse the brain and, if you like, try to breathe life back into the brain. Here is the model following a decapitation and surgical isolation. We perfuse it with synthetic hemoglobin, and this is the first model. Here we can measure what goes into the brain and what comes out of the brain. It's not connected to anything else, so we can calculate the rate exchange kinetics of just the brain without any contamination from any other organs. This is the original model. And here I'm sampling blood across the brain during different experiments. We can manipulate oxygen to it. We can actually float the brain, so we can alter the mechanics, the biofluidic mechanics in the brain. And again, we can do our very sophisticated measurements. We can take slices histologically and look at the brain. And what's fascinating is that despite decapitation, and this is roughly 8 hours post-decapitation, with reperfusion, we can see aspects of brain function coming back, both in terms of neurogenesis, gliogenesis, and synaptogenesis. This is the newest model now, several iterations on from the original model. This is a Parkinsonian brain, and we've now moved to humans in order to try to understand brain function, if you like, in a more detailed fashion. And we know that we can recover a great deal of function in the brain. Of course, clearly it's not the same as the brain before. And these are all fully approved experiments, of course. Not perfused and perfused, we can see a whole range of proteins, neuroprotective proteins that are being expressed in the perfused brain. And we can see a degree of recovery across all structures in the brain. And we can actually measure flow, we can measure oxygen consumption, glucose consumption, lactate formation, etc. So this is a really interesting model. And our long-term goal here, using this in vitro pulsatile, ex vivo pulsatile model, is to be able to digitize the brain so that we can fully understand how the brain works. Single-cell RNA-seq, spatial transcriptomics. We could look at different levels of the cell without geographically altering for sampling, and expose the brain to hypoxia, gravitational shifts, etc. The aim is to be able to have some really interesting targeted biomarkers of responses to environmental stresses, but also, of course, disease, neurodegeneration in particular. And it speaks in some ways to almost a hidden potential, a hidden energy reserve that we think potentially the closest we can get to this, of course, is in the free divers, where they can actually hold their breath for longer, perhaps than some of the mathematical calculations of oxygen volume depletion would suggest. And these are some experiments, unique experiments following approval from the family in terminally ill patients that with EEGs, they were still instrumented following extubation. And roughly 10 to 12 minutes following formal brain death, we see these terminal spreading depolarizations, cortical depolarizations, or brain tsunamis, almost as if the brain has gone into a cryogenic state and then, like a battery, is then starting to fire back up again. So really very interesting. And the work that we're doing at Bexorg again speaks to that, that there is some degree of function that can be recovered from the brain. And there are hidden oxygen reserves, we think, that we can't currently measure, but we think are aligned to this recovery of function. So it could explain some of the really extreme examples of cardiac arrest in cold conditions where individuals, patients have recovered without any apparent neurological deficit and against all odds, certainly against all bioenergetic calculations in terms of brain survival. So in terms of space, this has taught us many, many things. We've got an eye at the moment on this planet. This is Mars for deep space exploration. 0.376 G, a third of the G that you and I have grown up with, 1,000 days minimum return journey, depending on orbital position. Excuse me. And exposure to what we call the space exposome, altered gravity, radiation, isolation, distance, and it's the singular most hostile environment, near as darn it to a perfect vacuum in space. And this is a cause for consternation amongst many physiologists. And we're going back to the moon in order to establish a habitat for a future horizon goal of a deep space class exploration mission to Mars. And the exposome impacts every single organ. And so it's a wonderful opportunity for physiologists to get involved to try to understand the integrative impact that the space exposome has on the body. And of course, my focus is on the cerebral and the ophthalmological in this setting. No more important was this, of course, with Butch Wilmore and Sonny Williams. And I'm sure you saw this on TV when they were rescued after being stuck on the International Space Station. They were there for a week, but they ended up there for about 9 months. So they were fully exposed to the space exposome, and trying to decode the space integrome is a fascinating area of physiology because it involves multi-stressor dynamics. How do we model the change of gravity, space radiation, isolation, confinement? How do we model all of these stresses in a singular system, in a singular analog, in order to understand the integromic stress on the astronaut? Is it additive? Probably not. I'm sure it's synergistic, some evidence to suggest that. But it poses many challenges for us to really fully understand the physiological risk and impact of a deep space exploration class mission. So this is a very fertile area of research, and one that I think is wonderful for early career researchers to contribute to try to understand. There are very many exciting models that allow us to look at some of these stresses. And again, traditionally, most of these stresses have been looked at in a unicentric focus, just focusing on the singular stressor. But we need models where we combine multiple stresses simultaneously. This is a parabolic flight. It's a wonderful experience. Here you can see me spinning blood. When you pull gravity away from the brain, microgravity, we see overperfusion, if you like, in the posterior circulation and damage to the neurovascular unit. And this is perhaps a mechanism that we're keen to interrogate for a particular syndrome known as space-associated neuro-ophthalmological syndrome. We can do less expensive experiments, tilting people in the lab, exposing them to hypoxia, short-arm centrifuges. Skydiving is a wonderful, cheap model. in order to try to understand how freefall impacts cerebral bioenergetics, perhaps as a novel space analog, certainly cheaper. But what we are understanding on the space station, every time you look up in the sky, each and every one of the astronauts on the space station is engaging in between 1.5 to 2 hours of exercise. It may be good for the muscles, for the bones, for the heart, for the circulation, but we're not entirely convinced it's good for the brain. As you can see here, after 180 days on the International Space Station, we see some degree of compression, floating brain in space, and some compression. And we think that this could be a contributing factor, if you like, to space-associated neuroophthalmological syndrome, where we have these irreversible visual deficits, which is a red risk rated risk factor, if you like, for a deep space exploration class mission. We've just come back from an experiment with SpaceX. This is frame 2, so the first polar orbital mission with 2 males and 2 females. Wang was the commander here, the Chinese billionaire. We were interested in running an omics-based study, salivary and bloodborne, to try to understand the sex differences associated with spaceflight. That was a particularly exciting and a very novel mission. Spaceflight is now being commercialized, of course, and some wonderful opportunities for our early career researchers to engage with. Space countermeasures. How can we make space work? In other words, how can we survive deep space class explorations? And I've just stood down as the chair of the Life Sciences Working Group, just rotated out of the European Space Agency, and it was a wonderful experience where you can actually put some of your scientific interests to the test. Here, there's a whole bunch of colleagues from all of the 22 member states, different specialists in different fields of life sciences. And we were tasked with contributing to some of the high-level documents associated with the European Space Agency, Terra Novae 2030 plus, where we're interested in trying to focus research priorities and trying to make deep space flight as safe as possible. And one of the areas that I was particularly focused on is the integrative and countermeasures approach. In other words, how can we de-risk the space environment so that we can survive and thrive? And this allows us to, with colleagues, to publish consensus white papers in order to try to direct future research and to highlight the knowledge gaps and what we need to do over the next decade or so prior to a Mars crewed mission in order to make it as safe as possible for our astronauts. Absolutely fascinating bit of work. I really thoroughly enjoyed this, and working with 22 member states is just really great fun. From selection and screening to monitoring and support, it's a wonderful way to intersect with industry because they're really keen to fly super lightweight, really novel technology to make measurements in very remote, challenging conditions. And perhaps for the genetic specialists, future selection of Homo galactus in order to make deep space flight as optimal as possible. And I think smaller women are perfectly poised for a number of physiological reasons to be the ideal candidates for a Mars crewed mission. The systemic countermeasures from radiation shielding, personalized medicine, psychological support, nutritional support. How do you feed astronauts on a 1,000-day journey? These are all great questions. And what is the best exercise intervention? One size certainly doesn't fit all. As I mentioned earlier on, potential elevations in intracranial pressure could be a risk factor for the brain. The increase in flow is great for muscles, heart, and the lungs, but it could be a factor that could contribute to space-associated neuroophthalmological syndrome. There are some really novel interventions, for example, inducing torpor, hibernation, which we can do, of course, in warm-blooded mammals. But this is now becoming science fact, not just science fiction, in order to reduce oxygen consumption, perhaps provide a degree of neuroprotection so that we can optimize the health and well-being of astronauts. And this is a funded program at the moment with the European Space Agency. So some really great research going on in this area. There are other countermeasures, for example, lower body negative pressure. As I showed you earlier on, there's this headward shift, about 2 liters, cephalid shift of fluid towards the brain, potential for overperfusion and damage to the neurovascular unit. So sucking blood away from the brain is a really interesting countermeasure that we think can reduce intracranial pressure. It's still a divided question as to whether or not intracranial hypertension is contributing to SANS, but nonetheless, it's a very exciting countermeasure. And then other tiny little species. This is the tardigrade. Tardigrades you can freeze, you can boil, you can shoot them out of a gun, and they still survive. You can expose them to space. So what is it about their adaptive responses to these extreme environments that we humans can learn from? So just back to reality quickly, some high-level findings. Those are some of the models, some of the platforms, some of the interests, some of the applications of the work that we're interested in. What we've shown is that when we really drive a reduction in oxygen delivery to the brain in terms of arterial oxygen content, we see this reciprocal increase in blood flow. So this is an evolutionary conserved example of hypoxic vasodilation. And we maintain oxygen delivery to the brain despite real extremes of reductions in arterial oxygen content. So oxygen, as I mentioned, is really, really fundamentally important for the brain, and the brain is doing everything it can in order to preserve cerebral oxygen homeostasis. Even in those models where we can drive such extreme reductions in the arterial PO2, such that at the end of a breath hold, this is original data from Chris Willey, at the end of a breath hold, we have no arterial jugular venous oxygen gradient. The only way oxygen can then shift into the brain is by flowing more. And we do see this increase in flow. And many of our studies are showing that it's the posterior circulation, which is phylogenetically the oldest part of the brain and subserves the most delicate parts of the brain stem, the cerebellum, the medulla oblongata. And this seems to be preferentially preserved in these extremes of oxygen deprivation, these extremes of hypoxia. Radicals are generated as well, and for the early career researchers, stick with an idea. This is something that I had an idea back in 1997. It took about 8 years to be able to publish the first papers in the human model to demonstrate that with less oxygen, i.e. hypoxia, we see an increase in oxygen-centered free radical formation. So it seems almost counterintuitive, but now, of course, we know it's very clearly the case. Here you can see the arterial, this is an example of an arterial spin adduct. This is an oxygen-centered alkoxyl radical. The arterial circulation, the venous circulation, no change in the amplitude at rest, a minor AV difference such that the brain is generating a low-grade level of free radicals. But when we expose the individual to 12% oxygen, which is roughly about 4,500 meters exposure to acute altitude, so quite significant, we see an increase in the arterial concentration of radicals that is further compounded across the cerebral circulation in the jugular venous effluent. So we are confident that the brain is contributing markedly to the systemic accumulation of free radicals. We see this during gravitational exchanges as well, during some recent experiments in tilt, the hydrostatic challenges posed by tilt, and we can use ever more sophisticated spin traps and spin probes to look at different types and different species of radical generated, and how this intersects with nitric oxide bioavailability, which is tasting oxygen as the red blood cell moves through the circulation and drives these tonic changes that influence substrate delivery in terms of glucose and oxygen to the brain. So it's this delicate balance, if you like, this yin and the yang between radicals being generated, the different types of radicals, their reactivity, and nitric oxide bioavailability. An overperfusion of the brain, of course, can cause problems. As you can see here, this is um, um, a high susceptibility weighted sequence demonstrating what we think is red blood cell deposition in brain parenchyma as a result of too aggressive hypoxic vasodilation. And this is in patients with high altitude cerebral edema that really vasodilate very, very aggressively because they're profoundly hypoxemic. So we're still trying to understand what the optimal levels of hypoxic vasodilation are. What is the optimal balance in order to preserve the neurovascular unit, to preserve the blood-brain and the blood-CSF barriers. And we know that radicals are they're good. They're not just simply bad or plain ugly. This is an experiment that I did in collaboration with Russ Richardson at UCSD when I was postdocing. This is at rest and during maximal exercise. Here, prophylactically, we would knock radicals down with an oral antioxidant cocktail before they then came into exercise. And you can see we can profoundly reduce the generation of radicals. We did this in a forearm model where we're working the forearm. And what we found was fascinating quite some time ago is that when we knocked the radicals out, we saw more than a 50% reduction in blood flow to the forearm, brachial arterial vasodilation. So that's suggesting to us that we need radicals in physiologically controlled amounts. They are hormetic, they are acting as redox switches. But when it transitions beyond that threshold boundary to cause pathological damage in excess, then it becomes neuro maladaptive, if you like. And we've repeated a whole slew of experiments that we're still yet to publish from, where we infuse intra-arterially high-dose ascorbate, which is the most potent water-soluble chain-breaking antioxidant, to demonstrate that we are actually seeing a reduction in oxygen-sensitive gene expression that is involved, again, to suggest that these radicals in controlled physiological amounts are acting as hormetic molecular switches. And again, these radicals can act as quantum triggers. Erwin Schrödinger, it can act both as particle and a wave, and it can switch on a whole host of oxygen-sensitive genes that we might not fully understand by taking classical physiological approaches to understand these mechanisms, that we need to shift to something really quite different in order to understand their quantum effects. And this is very ripe fertile ground for research, trying to design experiments to demonstrate the quantum bases of signaling and driving neuroprotection. And it's this fine balance again that I mentioned, which is important for the neurovascular unit. And at the moment, we're also involved in the synthesis of novel nitrones that are mitochondrially target to try to trap some of the radicals in the mitochondria, which we think are the primary proximal sensors of changes in oxygen concentration. So sometimes research might feel a little bit like this. It's great fun. It's a real adventure. The G.L. Brown Prize has kind of helped me reflect a little bit on what G.L. Brown would always reflect on with his students. Very pedagogic. The work that we do, I think, has to be really encouraging, supportive, collaborative, integrative, and inspirational, I guess, in many ways. And these are just three, I guess, pearls of wisdom, dare I say, for the early career researchers that are tuning in. First and foremost, cultivate intellectual range, thrive on integration, and don't silo yourself. Be multilingual across systems, methods, and scales. Use a whole range of techniques. Working with physicists, mathematicians, physical chemists, geochemists, clinicians, it will enrich your understanding of the science. Learn to fail with speed, humility, and purpose. Experiments collapse, grants get rejected, equipment refuses to cooperate regularly. Physiology rewards resilience as much as brilliance. So just as we are resilient in environmental stress and as resilient as we can be for deep space exploration, you need to be resilient in terms of the research that you conduct as well. And you need to cope with failure. That's been a big part of my career, is failure. And it's an important part of coping with that and then super compensating and pushing on. And build your scientific tribe, protect it fiercely. Physiology is a team sport, and I'd really encourage you all to collaborate. The networks that you build across disciplines, countries, and generations, they matter way more than just a single project. And I think that's one of the greatest things about the work I do is this opportunity to collaborate with a lot of really great collaborators that have all become great friends as well. And with that, I'd like to thank you for your attention. Really appreciate your time. And it's been a pleasure speaking to you this afternoon.
Thank you so much, Damien. That's brilliant. So we've got a few minutes for questions if there are any. I'm just checking. I don't think there's any currently in the chat, so please do put them down there. I've got loads though, so I can like start us off. So one thing I just wanted to ask on a like, what do you think's going on in the brain of a free diver who's say been breath holding for 5 minutes and they've got another 5 minutes and they're not gonna like what are their neurons doing that means that it's okay that they've only got like they've basically there isn't any oxygen availability barely. So how come they've not they're not having anoxic depolarizations and things like that.
Well, I mean, one of the things that we don't um, assume, of course, is that we don't assume that we're not seeing evidence of brain damage or altered reactivity in the brain. So we did a study, we published it quite recently in the journal Cerebral Blood Flow and Metabolism, where we looked at Samoa Quanterix. We looked at Neuroplex 4 platform of brain-specific proteins that would suggest damage to the neurovascular unit, and demonstrated that during a breath hold, which is a combination, of course, of very low oxygen, which drives vasodilation, and very high CO2, which drives vasodilation. CO2, of course, being more of a potent vasodilatory stimulus than the hypoxia, to demonstrate that we saw the release of brain-specific proteins that would suggest an increased reactivity in the neurovascular unit and potential disruption of the blood-brain barrier.
Mm.
Uh, and that would make intuitive sense to me because, elevations potentially in intracranial pressure, huge elevations in blood flow. There's such a double whammy of a hypoxic, hypercapnic vasodilatory stimulus can cause damage to a very sensitive, vulnerable, fragile neurovascular unit. And I know colleagues are chasing this at the moment to look at this is quite a novel model of intermittent, repetitive, recurrent blood-brain barrier disruption and then corresponding susceptibility to later life neurodegeneration. So, it's not all good for the brain. These athletes are amazing athletes. They're a phenomenal group that are pushing their brains to the extremes. And when you watch somebody that's breath holding for longer than 10 minutes, and certainly those that are on hyperoxic gas for 15 minutes and can extend it to almost 25 minutes, it's fascinating. And
But
a
what
lot
are
of
they
the work.
thinking? Sorry to interrupt, but
Yeah.
like, what are they? What I mean, so I totally get that. That like, it's probably not. It's probably doing, like you say, causing damage that probably would add up in the long term. But what at that moment, when their neurons aren't able to, presumably aren't able to produce enough ATP to function normally, how do they report? Do they report what they think? Like when you talk to them about what they're thinking when they're like halfway through that breath hold, what do they say they're thinking about? Like, what's their cognition like at that point?
But what's really interesting is that in these really extremely adapted athletes, these top end athletes, and working with colleagues at UBC, Phil Ainsley and Tony Bain and Ryan Hoyland, initiated by Chris Willey, by the way. What's fascinating about these athletes is that with the hypercapnic hypoxemia, we see a reduction in CMRO2. So we see over the duration of the breath hold, a reduction in the cerebral metabolic rate of oxygen. So rather than a panic, anxiety driven elevation, if you like, in oxygen consumption, we see something that's not too different to what we would typically observe in, for example, a turtle. That reduction, almost a cryogenic state, which is neuroprotective, reducing oxygen consumption, is probably neuroprotective. But they defend oxygen and substrate delivery to the brain. So they're pushing the brain extraordinarily hard, but yet there seems to be sufficient substrate in order for them to continue to function. But the architecture of the neurovascular unit, I'm absolutely convinced, is being disrupted. It's being
Yeah.
challenged, really very aggressively, just as we would see with a patient with high altitude cerebral edema. This marked, and it would be really interesting to do some imaging here in order to look at the blood-brain barrier, to do some labeling, to look at the barrier disruption, kind of, temporarily, over the course of the breath hold. But yeah, you're right, Catherine. They're a fascinating group of athletes.
Yeah, it's amazing because you can ask them, you can't ask turtles. So it's like it's
Yeah.
a great resource. Sorry, there's a question in the chat about the mechanisms behind autistic swimmers. Do you mean not and might have to get the questioner to add a little context. Are the mechanisms behind autistic swimmers the same as the ones in the free diver? So is that maybe that,
The breath holding.
yeah, so but I guess they're having to then do coordinate activity, like, complicated movements at the same time rather than not having to move. Right, yes, that's
Yeah, it's a really interesting question, really. I think the synchronized swimmers, if you like, the artistic swimming, you've got a double whammy there in terms of challenges for the brain. Well, actually, a triple whammy in as much as you've got exercise, you've got the hypoxia, you've got the hypercapnia, and I have watched the synchronized swimmers. I'm not a specialist in synchronized swimming by any stretch of the imagination, but you can imagine greater hypoxemia. You become more diffusion limited, of course, and more hypercapnia as a result of the physical activity. And all of these things can impact cognitive function. What we do know, what unifies this age-related reduction in cognitive function, and why dementia is more an age of the older brain, of course, there are exceptions, but is it's unified by this reduction in cerebral blood flow over the lifespan. Um, and they're performing quite cognitively difficult, challenging tasks, and the brain is under incredible pressure that would be suggesting that cognitive function is likely impaired. So yeah, it's they're a great model. I think we would probably struggle to convince synchronized swimmers to have a jugular venous catheter or an arterial catheter placed, but certainly something that's worth considering.
brilliant. Thank you. So I also I've one more question maybe, unless there's others here. Because why do you think the posterior circulation is more sensitive? So what's different or special or more less resilient about the posterior circulation?
So, I mean, the posterior, so if you look at the brainstem, the cerebellum, the medulla oblongata, phylogenetically, this is the most evolutionary conserved part of our brain. It's the oldest part of our brain. What makes us human, if you like, the frontal circulation, the cortical circulation, it's jam-packed full of tissue and it's very expensive to run. It's about 12 to 15 times more expensive to run than any other somatic tissues. The brain is, as you know, very, very oxygen hungry, almost over-engineered in many ways. So when you look at the posterior circulation compared to the anterior circulation, there are clear differences, for example, in terms of CO2 vasoreactivity. There are differences in dynamic cerebral autoregulation. There are differences in perfusion, hypoxic sensitivities that likely converge. I mean, it's not absolutely black and white, but if I was to put an opinion on it, I would say likely converge to suggest that the more important parts of the brain, if you like, involved in autonomic function and the maintenance of acid-base balance are probably the most well preserved, if you like, when under pressure. And these extreme models of physiology are really putting the brain under pressure. And it's then and only then when we put them under pressure can we start to see a differentiation, a regional heterogeneity of physiological response, if you like. And what I find fascinating is that you look at arteries that supply blood flow to the brain in the neck, the extracranial arteries, and those inside the skull, the intracranial arteries. The heterogeneity of flow and vasoreactivity is fascinating. Not one single artery behaves like another. And again, there's clear reasons for that. And there's been some wonderful work looking at this spatial regional heterogeneity, again, as I mentioned, coming out of UBC, looking at duplex ultrasound. Again, Chris Willey, the late Chris Willey, was instrumental in a lot of this work with Phil Ainsley. So, yeah, fascinating.
And so for that, so it's the posterior circulation that's preserved in conditions of, let's say, reduced oxygen availability, but then conversely, when you've got low gravity, that means that's the bit that then sees the highest increases in flow and then the space related damage. Is that correct?
Yeah, and of course muscle can flow really hard and you can get away with it. The brain doesn't like to flow hard. And what's even more fascinating is that it only extracts 30% of the oxygen that it sees. Why isn't it not extracting a greater percentage of the oxygen it sees and then throttling back, if you like, in terms of perfusion? So, these are all really interesting bioenergetic questions. But in the situation where we expose the brain to microgravity, for example, parabolic flight, where we're seeing a more vasoreactive response to the microgravity in the posterior circulation. So the brain is fighting to maintain blood flow to itself. We're actually seeing a downside of that, which is overperfusion and then mechanical disruption, we think, then of the neurovascular unit, which could be damaging. In a situation like that, we've got some protein-based work, not imaging work, but protein-based work to suggest that we think the blood-brain barrier is being disrupted. And this could contribute to an extracellular vasogenic edema, if you like, a buildup of water in the brain, which we see in hypoxia, by the way, as a result of the hypoxic vasodilation. And that could then result in brain swelling, that extra volume overload, depending on whether or not there's sufficient venous outflow. We think there's a degree of obstruction there as well. This can cause an elevation in intracranial pressure. And it's that intracranial pressure increase then that can impinge, compress the optic nerve to cause these ophthalmological deficits. So, it's not absolutely clear if it is intracranial hypertension that's driving this syndrome, but I'm pretty convinced that there's a contribution, at least part of this is mechanistically important.
Thank you. There's a question. Jeff Coombs has his hand up. We've asked for questions in the chat, but I'll make an exception if you can ask it quickly because we're pretty much at the end of the session. If Damien's all right, are you all right with
Yeah,
one?
of course. I know Jeff well, so hopefully he'll
Okay.
ask a nice question.
Even though he's not followed the rules. That's all right, go for it.
Oh, Jeff, you're on mute, I think. Oh,
Can you hear me, Damien?
oh, that's better. Yeah. Hey, Jeff.
Okay. Hey, Damien. Sorry, I'll try to make it quick. Thanks for
That's
the really
okay.
interesting talk. We have a few students here who might be interested in hearing about how the ultrasound-based methods that they do in the lab kind of transfer to the microgravity environment. I don't know if you can talk about that quickly, whether it's TCD or carotid scanning, if you have any insights on that.
Yeah, thanks, Jeff. So, uh, really interesting in terms of so we've used duplex. So the work that we've published that I was mentioning to Catherine there in terms of the overperfusion in the posterior circulation, there we were measuring, anterior flow through the internal carotid artery and posterior flow through the vertebral artery, and then intracranial flow frontal anterior through the middle cerebral artery. We did that with duplex, but we've also used transcranial Doppler, which, as you know, focuses unicentrically on velocity, not flow. The reason we did that is so that we could look at dynamic cerebral autoregulation during these gravitational transitions. So it's really fun, actually, during duplex. What you don't do at sea level is you don't attach yourself to the floor with a carabiner and rope when you're doing duplex, but during the gravitational transitions, the parabolic flight is, the microgravity phase is flanked by a hypergravity phase, 2Gs as you move into the parabola and 2Gs as you come out of the parabola. So you're being pushed into the ground, double your body weight. But as you hit that 25-second microgravity phase, if you're not clipped into the carabiner, you're going to end up on the roof of the airplane. And that happened a couple of times. It's not easy to get into the vessel. So, we try our best to insulate the vessels, but there is a degree of, inaccuracy associated with that. But of course, repeated measures and statistics, the laws of probability help us get around that to some extent.
Thank you. Right, so I think we should probably call it a day. We've overrun, but it's been super interesting, so I think that's great. So thank you everyone for attending. Thank you, Damian, for giving us such a great talk. And thank you for attending and participating. Bye, everyone.
Pleasure. Take care. Bye-bye. Thanks, everyone.
Thanks.